Dear ladies and gentlemen, dear colleagues, I assume we're live. It's a pleasure and an honor to welcome you to this week's ESDR Kitchen live webinar. It's going to be a sweet and sour. Two excellent speakers will cross their blades. Actually, they started hitting back and forth already in the room before that. So it's going to be, I assume, a funny and entertaining discussion. So we have in the right corner, we have David Adams. He's the head of the Experimental Cancer Genetics Wellcome Trust Sanger Institute in Cambridge in UK. He actually did his PhD in Sydney and now is obviously focusing on the genetics. And he's also the co-head of the Cambridge Cancer Center Cell and Molecular Biology program. He's a fellow of the Academy of Medical Science and the Royal College of Pathologists. He is also a distance runner up to half a marathon. So we'll see if that helps him in his infight later on. In the other corner, we have Neil Rajan. He's MD PhD. He's a dermatologist. He's a principal investigator, and with his group, he's really focusing on translational research, particularly in rare genetic skin diseases. He has several awards for his work, which was published, among others, in Nature Communications and Science. Not sure if he's boxing or fencing or anything else, but we'll see how he managed in this fight. So I think David will start first. So the ring is yours. Just to let you know, before we start, this episode is sponsored by Pfizer, which enables us to provide these episodes. And it's going to be on YouTube in 1 or 2 days. And please, if you have questions, please put them in the Q&A or in the discussion. If you have time in the end, we'll put those questions up to Neil and Adam. And David. So David, the stage is yours. I mute myself. Thanks a lot.
Curdin, just
you're going to first. You're going to first. Sorry about that.
No, not a problem. Thank you for that kind introduction. And I'm hoping you can see my slides. So thank you, everyone, for inviting me to this great opportunity to speak at this debate. And I'd like to introduce the topic a little bit today. When Dave and I were coming up with titles for our debate, we wanted to be provocative, and that was to garner interest in the area of skin tumor sequencing. And really, we wanted that interest to inform discussion and dialogue around how we could best do this. And so perhaps the first spoiler alert is, despite the fact that I'm fighting for the sour side or the side against this, I'm a huge fan of skin sequencing, and I hope that the messages that we come out of this exercise today will improve the quality of that for research generally in skin. So my conflicts of interest slide, I've got no relevant conflicts to declare, and also a range of sequencing technologies are available beyond those in this talk. I wanted to speak to these outcomes, and really when researchers are embarking on a project that draws on sequencing skin tumors, there are a number of optimal outcomes, much like across other skin cancers and cancers in general. So we ideally want to find a novel insight into tumor biology that DNA can tell us, and that this is specific to a tumor type or stage. We ideally find a clinical benefit for patients with a translational application as a diagnostic or prognostic biomarker with this finding. We can potentially use such a finding for therapy and personalization of cancer care. And in some families where there are predisposed individuals who develop many tumors, these may lead to preventative therapies. However, despite these overarching aims, we all know of sequencing studies in skin tumors where, despite wanting to achieve that, that's not happened. And over the course of the next 10 minutes, what I would like to highlight are pitfalls in study design, which I think may have accounted for some of those things. And ideally, by recognizing these pitfalls in advance going forward, we're able to move around these challenges and get better outcomes. So in outline, I'll say a little bit about genetic sequencing. I will then speak about the importance of good clinical phenotyping and how crucial dermatopathology is. I will say a bit about the selection of cellular material for sequencing, what goes in, and also which controls we might want to use and choose. Finally, the role of data analysis and interpretation, and the importance of having sufficient numbers of samples of that tumor type for validating and generalizing these results. So genetic sequencing has come a long way, and I really like this slide highlighting the Human Genome Project's 30th birthday. And it puts it into the context of Gregor Mendel and the Peas back in 1865, and kind of brings us forward to a time when Fred Sanger was credited with Sanger sequencing in 1977. And really over the last 30 years, huge steps forward have been made with seminal discoveries that ultimately in around 2003 allowed us to have a reference. And so if you're a cancer biologist, this was an exciting time because now you could compare things against this reference and ideally find things that were different in cancer. And this is on a backdrop of the cost of the human genome sequencing dropping over that time. And we're now in the realm of $1,000 genome, as you'll see here, at least from a sequencing cost alone. And sequencing is a technology which I describe as a classic GIGO, or garbage in, garbage out. And so if you are very careful with what you put in and very careful with your experimental design, you stand to gain a lot. But if you don't take those steps, then there is the potential to be faced with results which are less useful. And sequencing is happening across the planet at scale at the moment. There are 41 countries with national genomic sequencing projects. And of these, in 16 countries, it's shaping education, all the way from primary school up to tertiary education. And so whether we like it or not, sequencing is happening, and it's increasingly a part of our healthcare. And from a cancer perspective, the Cancer Genome Project has one of its outputs as a catalogue of somatic mutations in cancer, and it sort of gives a scale of how far things have come along. Now, this is a nice infographic from 2019, but in release 94 in May of this year, there are about 1.5 million cancer samples that have been sequenced, and of these, about 38,000 whole genomes. And so a lot of this is already happening. The technology that is used currently is largely next generation sequencing, or as I call it, sequencing. And there are two main technologies, and predominantly a short read technology has dominated the market. This usually gives you between 75 to 150 base pair reads of nucleic acids. And it is an accessible form of sequencing. There is a low base error rate. These graphics here sort of highlight how low that rate is. And it is perhaps compared against emerging technologies that are increasingly being implemented in genomic studies, which use long reads between 10 to 25,000 bases. And these long read technologies can give you different insights and can now give coverage over regions of the genome which were harder to align, such as highly repetitive regions. It's important to recognize when looking back at sequencing experiments that were a waste of time or not, that not all sequencing is the same. And over that time course, which I showed you earlier, experiments have been done which might have focused on single hotspots, which have given tremendous insight and clinical value, such as in BRAF, or sample experiments which have looked at important genes, such as P53. Over a period of time, many of these approaches have been expanded to include targeted panels of cancer genes, coding exomes, and more recently, whole genomes. Also, sometimes people sequence RNA, and this can allow the analysis to incorporate fusion transcripts at lower cost than whole genome sequencing can, for example, and sometimes can be really useful in looking at the impact of a single of a variation in the DNA and its impact on gene expression or isoform expression. So I've said a bit about the sequencing, but what do we sequence? And here, as a clinical dermatologist, these are images of squamous cell carcinoma. It is one thing, I guess, to call this a squamous cell carcinoma, but I think it is crucial to recognize elements such as site. I mean, we increasingly realize that across human skin, all regions are different, and we know that at certain sites, certain cancers of the same type have a worse prognosis. And really, tumor location, even beyond sun-protected and sun-exposed, is key in terms of phenotypic variables to have for the data interpretation. Also, the patient phenotype is important. For example, is the patient immunosuppressed? Is the patient a transplant recipient receiving therapies such as azathioprine, where mutational signatures specific to that have been shown recently? Is the patient HPV vaccinated, or does the patient have a genetic condition that may predispose them to certain rare types of squamous cell carcinoma? And here there are images of patients with xeroderma pigmentosum and recessive dystrophic epidermolysis bullosa. Also, dermatopathology is a specialty which is really super specialized, and these samples here by some may have been labeled as types of squamous cell carcinoma, but are in fact acrine porocarcinoma. And certainly there are different clinical patterns that can have certain clinical phenotypes, and these need to be characterized by a dermatopathologist that's seeing lots of these rare cases when collecting them for your sequencing experiment. Also, some dermatopathologists may use immunohistochemical markers to further categorize these tumors. And this is important because as the heterogeneity becomes apparent, one can then refine the number of samples that are needed. In terms of sample quality, there is the key question of which controls do you use? And this seminal paper by Inigo Martin Carina with Mike Stratton, Phil Jones, and Peter Campbell was really important in highlighting that even in normal eyelid skin, there are mutations that are present that are recognized in a range of cancers. And so there is a question of, do we include controls just in the skin, or do we look at other tissues such as blood and saliva, for example? There is increasingly a drive to look at normal cells and the mutational load of those, and this paper led by Hunter Shane and Boris Bastian looks at the mutational burden in single normal melanocytes from a range of different sites. And you can see that in some of these melanomas, there are in the order of 30 mutations per megabase in normal melanocytes, not nevi, not melanoma. There is also the broader context of how malignant drivers that we find in, say, melanoma or SCC sit in the context of benign lesions. And whilst this has been done in some lesions, such as nevi, there are about 70 to 80 different benign skin tumors in the WHO categorization, and many of these have not been sequenced or studied. And it may be that many of the findings that we're linking to cancers are present in benign lesions, and this is an important context to interpret this in. In studies where samples have been studied from the level of normal skin, benign precursor, and melanoma, this has led to interesting insights into tumor evolution and acquisition of driver mutations. Finally, the type of samples that are used are critical, and we're largely in a space where FFPE samples or paraffin-embedded samples are most accessible, but it needs to be recognized that fresh samples can bring new insights, both in terms of quality of RNA, should RNA sequencing be done, and in terms of the long-read technologies, these will offer DNA which is non-fragmented, and so can influence what could be found in those studies. Tumor cellularity is something that is a real bugbear, and it can be incredibly hard to quantify. And there is a scenario as we move forward where machine learning approaches are now being used to categorize regions of tumors and count the levels of cellularity. So all of these steps will improve yield in future studies. A further contemplation to bear in mind is that for many sequencing experiments of cancers, so far it is estimated about 0.0005% of a cancer has been sequenced, and really we might be missing out on the bigger picture. And there have been moves afoot to look at homogenizing whole tumors, which are not needed by the pathologist, and studying those materials that might give a better comprehensive picture of drivers in a cancer. I will say a few final points. One, that over the period of time that sequencing has been happening, our ability to detect different variants has not been equal. And certainly in terms of single nucleotide variants and indels, some of these technologies have been excellent at detecting that. But as we go forward, we'll be better placed with whole genome and long read technologies to look at structural variation. When we come to the actual mutations, it needs to be recognized that skin is highly loaded with mutations, and melanoma sits in pride of place at the top end. And even recently, actinic keratosis and squamous cell carcinoma have nudged that out of the way. And so there will be many variants in a range of different skin cancer sequencing experiments. I will finish by saying that this is where excellent bioinformatics is key. And by using the best packages in tandem, one can potentially go from tumor-specific somatic mutations through to variant filtering, discovering significantly mutated driver genes, and coupling this with other elements such as copy number changes, translocations, and fusions. And more recently, of course, mutational signatures have been a further element that has provided insight. So I'll stop there, but I'll summarize to say it is easy to sequence, but it is harder to make sense of sequencing data. That experimental design is key, really, to get the most out of sequencing. Otherwise, there is the risk that we could be wasting time, and that the clinical context is also important for overall interpretation. And with that, I will stop, and I will hand over to my counter, David, who will talk about the sweet side of sequencing. Thank you.
Thanks so much, Neil. Okay, can you see my slides okay?
I don't think you're sharing yet.
Not yet.
Okay, let me go again. Here we go. How about now?
Great.
Good.
Okay, thanks so much. So thanks so much to the ESCR for the opportunity to speak. I'm going to give you my perspective on the sequencing of skin tumours and whether or not this is a fruitful effort or indeed a waste of time. So these are my conflicts of interest. So I thought I might start here by talking about the MAP kinase pathway. So this is a pathway that's critically important in the proliferation of melanocytes. And more specifically, there's a gene in this pathway which is called the BRAF gene. This gene, by sequencing, has been determined to be mutated in a proportion of both melanomas, but also benign melanocytic nevi. And as Neil mentioned, the particular mutation that can be acquired in this context is called BRAF V600E. So by looking at the tumor or the melanoma or the nevi on the skin, it's really not possible to determine whether that particular lesion carries a BRAF V600E mutation, nor is it possible by simply looking at the histology of the case as well. So the best way of determining whether a case is BRAF V600E mutant is by using DNA sequencing. And you can see an example here of a tumor where there is a BRAF V600E change shown here, and this is the corresponding normal. So sequencing is the best way of determining whether or not there is an alteration. There are approaches such as immunohistochemistry that can be used, but importantly, there are other alterations that occur at that V600 codon, such as V600K and V600R, which are also clinically important. So why is this alteration or these alterations which have been identified by sequencing important? They're important because the sequencing led to the identification of a drug called vemurafenib. This drug can be used to treat patients who have mutations in BRAF at V600, at that V600 position. So this particular drug can be associated with profound clinical treatments, as you can see shown here. However, whilst a proportion of patients receive a fantastic response and in some cases cure, many patients who are treated with this agent will ultimately relapse on therapy. And here too, the sequencing has been very important. So the sequencing has taught us that the mechanisms of resistance that are operative in this context include reactivation of the MAP kinase pathway through amplification of BRAF or other alterations in BRAF, or indeed other alterations that affect the PI3 kinase pathway. So in the same way that sequencing can help you identify alterations such as driver mutations, it can also help tell you exactly which type of tumor you're looking at. So shown on this slide is an example of a cutaneous melanoma, and there's another case that is an acral melanoma. So just by looking at the pathology, it's not really possible to tell exactly which is which. However, if we look at the genomes of these particular cases, or indeed when we compare cutaneous melanoma on the skin versus, for example, acral melanoma on the hands and the feet, we can see that there are some differences. So on melanoma forming on sun-exposed sites, we can see there's a high mutational load with a particular preponderance of C to T mutations, whereas these alterations are largely absent from most acral melanomas. Conversely, we find that acral melanomas have a large number of copy number gains and also copy number losses. So these genomic features, these somatic genetic features, can tell us something about the paths towards tumorigenesis and the biology that is operative. So the other place where sequencing has been really important is in correlating germline changes together with the appearance of the tumor as it grows and develops. And just one good example of this is tumors, melanocytic tumors that develop in individuals who carry germline variants in the BAP1 gene, or the BRCA-associated protein 1 gene. The melanocytic lesions that form in these individuals have characteristic features, which include multinucleated melanocytes, but also intranuclear pseudo-inclusions. So the sequencing data has directed us towards a predisposition gene, and this is, of course, clinically important in terms of diagnosis, management, and organization of patients in the clinic. So skin is really the most amazing of organs. It protects us from the environment. In its texture and appearance, it tells us something about an individual's health. And as we age, it changes with us, and it also chronicles those past exposures. So because of this, together with investigators around the world, we started a project which is termed the Derm Atlas Project. And the aim of this project is to sequence tumors that arise within skin from all the different amazing cell types and structures that you can find. So this project uses sequencing to firstly identify driver genes, for example, such as BRAF V600E, to explore cells of origin by using bulk or indeed single cell sequencing, and also to explore the etiology of tumorigenesis. For example, to look at viral sequences, but also to explore the mutational processes that are operative. And as Neil described, these kinds of approaches, mutational signature analysis, can tell us about exposures or indeed predisposing alleles that might be important in the genesis of a cancer. We also sequence germlines, and in this way, we can try and identify predisposing alleles that might increase an individual's risk of developing the disease. So as part of this project, we're sequencing 70 different tumor types. This is 50 different tumors for each of these entities, and we're generating exome and also transcriptome data. In addition to capturing the genomic data on each case, we're also capturing an image, and this gives us an opportunity to use approaches like machine learning, but also to generate a resource to train the next generation of dermatopathologists. And the data we're generating is being released to the community, and these are the pathology leads for this project. So as an exemplar, what have we learned so far? So the first set of tumors that we analyzed were skin adnexal tumors, skin appendage tumors, and these include spiroadenomas and spiroadenocarcinomas. And so what we identified by sequencing these tumors was a hotspot mutation in a gene called ALPK1. And this appears to substitute for alterations that occur in SILD, which is another gene associated with these tumors. We've also been sequencing tumors such as sebaceous tumors like the one which is shown here, and we've been correlating features such as germline mismatch repair deficiency or the germline status of the patient together with the profile of mutations that we observe in the tumor. And we're also examining the potential role that viruses may play in the development of this disease. We've also been looking at rarer presentations of more common tumor types. So this is an example of trans-differentiated melanoma. So this is the conventional melanoma component here, and this is the trans-differentiated region here. So it's lost expression of melanocytic markers, but it's switched on expression of other markers such as Desmond. And we found that by sequencing these tumors that there was an increased frequency of NF1 mutations. So sequencing can tell you all of these things about the biology of these diseases. So these are the tumor types that we're sequencing as part of the Derm Atlas project, and they span the full spectrum from benign, intermediate, and malignant. Importantly, we're focusing on tumors in particular where there's very little genetic information available, as you can see shown here. So this is the number of genetic studies that have been performed across these different tumor types, and the tumors that we're sequencing are indicated here in red. So in the last few minutes, or the last minute or so of my talk, I really wanted to push the boat out and make a play for the idea that we shouldn't just be sequencing human tumors, but in fact, we should be sequencing tumors that arise in other species. And I want to do this in the context of mucosal melanoma. So this is a type of melanoma that forms on mucosal surfaces. It's associated with a relatively low mutational burden like acral melanoma, but it's associated with a large number of copy number alterations. These are the established driver mutations that have been identified within this tumor type. So this is a malignancy that not only affects humans, but it also affects other species, including dogs. And this is an example of a mucosal melanoma forming in the oral mucosa of a dog here. It's known to, in particular, develop in particular breeds of dog like Cocker Spaniels, German Shepherds, and also Boxers. And like humans, in this context, it's known to be highly metastatic. So this malignancy also forms in horses. And you can see an example here of one of these equine melanomas forming in the region around the lips. They also can form around the tail. But intriguingly, unlike humans and dogs, the tumors that form in horses are rarely metastatic and usually have a relatively indolent clinical course. So by sequencing tumors from each of these different species, what we can do is look at the shared genetic architecture, the somatic genetic architecture of these tumors, and this can really help us focus down on the key genetic events, the somatic alterations that are important in driving forward these tumors. And so we can see genes that are shown here that are commonly mutated across these different species, but also some differences. For example, this splicing factor here, which appears to be exclusively mutated in human and not in the other species. And this can really direct us towards some interesting and new biology. So the other place or the other approach that we can use is by looking at the larger scale copy number alterations that can occur within genomes. So this is the large-scale copy number changes that occur within human mucosal, dog mucosal, and also equine mucosal melanomas. And so by comparing between species, what we can do is find those regions of the genome that are recurrently altered in human, in dog, or in horse, and look at the shared genetic changes. And this can direct us towards those key genetic events that are important in these cancers. So rather than asking whether or not sequencing skin tumors is a waste of time, my response is that we should sequence everything. Because as I've illustrated, and as we've learned over the last few years, we can learn a great deal from this sequence data about the biology and the genesis of these conditions. I'd like to close by thanking the PIs of the Derm Atlas project, including my fellow speaker, Neil Rajan, and also the members of my group who've contributed greatly to this work, in particular, Louise van der Weyden and Ingrid Ferrara, and the MRC who have funded the work that I've spoken about today. Thanks very much.
So thank you very much. I think there's no questions so far. I think you wanted to shout back and forth a little bit.
Yeah. So Curdin, if I can start, and firstly, David, I think that was really nice, and I think the comparison across species is really fascinating. And one thing that struck me is this scenario where even in different species, there is a phenotype like melanoma, and the behavior can be relatively indolent. And what this sort of made me think about is, at this stage of sequencing, you highlighted how in some cancers we've done a lot of studies, but in many tumors we haven't, and we simply don't know what's there. But the next tier almost seems to be beyond looking at what's happening in terms of drivers in the tumor cells, what's happening in the surrounding microenvironment, what's happening in the matrix around those tumors. And I guess I was interested in how you saw the incremental building of knowledge from establishing what's happening at bulk first across a range of tumors, would then inform the design of experiments with more granularity, leveraging the newer single cell technologies.
Yeah, absolutely. So there's obviously spatial transcriptomics, which has come along, which has allowed us to look in detail at those the organization and structures, which may well play a role in the differences in presentation, not just within a species or within humans, but indeed within other species as well. I think there, as you were saying, really close associations with the dermatopathologists is critically important. They are key to all of these studies, as is high quality bioinformatics. And I think, bringing all of that together, consolidating that data is going to be what we're going to be focusing on for the next, decades.
Yeah. And I think I really like the fact that we can continue to link back to the clinic, because I think the other challenge that I've seen across the range of different studies I looked at when I was trying to determine where we had, made advances and perhaps were less successful is often in terms of the clinical phenotyping, there is sometimes limited follow-up data. So, and I think this isn't a fault of the researchers. I think this data is actually quite hard to get. And so when we see a melanoma in a patient and we have a snapshot of it at that time, we know what drivers are there, but then we sometimes don't know whether those drivers will link with a bad outcome or a good outcome. And I think there is scope on the front of the clinicians to provide better data, and for us to have better frameworks to capture that data, because really the interpretation of variance has to sit in that broader context. And yeah. I think it's asking for a lot, but I think patients expect a lot.
I think the role of the clinician in terms of collecting high quality data is grossly underrated. I think we need to work out better ways of incentivizing them to engage in these sorts of studies and collect data, because they're the ones who are with the patients, consenting them and so on.
And I think these are important agendas more broadly, because sometimes many of the exciting things that we want to do are often labeled as research, and getting that clinical support is not often costed into that research. To touch on it, there was a study recently that highlighted that although at the level of, say, $1,000 genome for families with genetic conditions, the actual clinical cost of delivering those genomes in a trio, for example, is in the order of about £5,000 to £6,000 in the NHS. And so those other invisible costs, I think, are some of the reasons why some of the prior efforts maybe haven't been as well resourced, and we sort of need to look at this going forward. But I'll stop there, Curdin. I appreciate their questions in the chat now.
Yeah, David, wanted you to add something?
No, I was gonna try and help with the questions briefly and just sort of try and wrap together Daniel and Sarah's question there about implementation, challenges for implementation. What do you see that they are, Neil?
So I think I've hinted at this a little bit. It has to do with thresholds for accessing tests. And in my clinical experience, I found that even 10 years ago, to get a genetic test, never mind in tumors, just in patients with genetic disease, would depend on the goodwill of a research lab somewhere that was happy to, help you out. And then this would feed back into a clinical result. Whereas in 2021, we now have a test directory and we can order panels. And in dermatology in England, certainly there is mainstreaming such that dermatologists are now ordering these tests, not just geneticists anymore. So I think the ability to access these tests are increasingly available. And I've hinted at the global scale of sequencing. Sarah's got a second question about the approach, the challenge of distinguishing pathological driver mutations from secondary effects on DNA integrity in malignant tumors. David, do you want to comment?
I can have a go at that. So 2 answers to that question. One is the mathematical answer. As Neil said, you need large numbers of tumors to really do good statistics. There's various mathematical approaches for doing that. Inigo and Martin Carino, one of my colleagues, developed a software called DNDSCV, which can tell you mathematically what is a driver. As a biologist, my response is, we should have, the next phase is generating cell lines or organoids or whatever from each of these different types of tumor and testing these things functionally. That's the level beyond. I think that that's going to be necessary to take the genetic data back to the biology. That's where we should go next.
So maybe to hook up on that a little bit, I mean, for the clinic, it would be simple, have these small panels that drive maybe a clinical decision making on what target, but for the research, does it make sense to just sequence everything, as you said, like, just the entire tumor? Or, wouldn't you just say, like, for the future, we should really go for single cell or at least stuff like DSP and to show the granularity of the tumor, particularly for tumors which have so much mutations in it, which are not all over the place identical?
I think you would probably argue that you need both, because there are some tumors, as I showed, where there's been no genetic data, and the starting point is to generate exome sequencing data. Every time I talk to Neil, he shows me an interesting case that I really want to sequence because it's got a different presentation, and I just want to get that initial snapshot about that tumor. Conversely, we're still learning a lot about cutaneous melanoma, where we've got hundreds and hundreds and hundreds of cases, and single cell sequencing and exploring that genetic landscape that Neil mentioned is still an area where we're learning things every day. So I think initially exome genome sequencing to get the landscape at a course level, and then go into the other approaches, single cell sequencing and landscaping with spatial transcriptomics.
Okay, so thanks a lot. We're coming to an end. We have to close. So thanks a lot to you, David and Adams. Just Brigitte Lane says the same. I think 2 excellent speeches and a very nice discussion. It was fun listening to you guys. So thanks a lot. We'll have the last 2 episodes of this year's ESDR Kitchen episodes, which will be on the 1st of December by Ellen van den Bogart on skin models and 3D epidermal skin models. And then there's another one, the last one, and I think mid of December will be the very last one. And again, as mentioned before, the question, everything, you can still add them on. We'll have them answered by the 2 speakers if needed. And in a few days, tomorrow or after tomorrow, the talk will be live, will be recorded and will be put onto the YouTube channel. So thanks a lot for everyone, and see you in 2 weeks. Bye-bye.
Bye.
Bye.